Welcome to The Long Run. This is a podcast for biotech adventurers. I'm your host, Luke Timmerman. Today's guest is Tim Lu. Tim is the co-founder and CEO of South San Francisco-based Senti Biosciences. Senti is working to develop gene circuits for cell therapies. The idea might sound like sci-fi to someone unfamiliar, but the company is working to reprogram cell therapies with precise genetic instructions on what to do in certain circumstances in the body. The genetic code essentially can tell the cell to kill tumor cells that bear a certain molecular marker while sparing other cells that carry a particular molecular signature. The first generation cell therapies have really delivered some extraordinary results for patients with cancer, but they also have some limitations. If Senti and others in the cell reprogramming world are successful, they could take cell therapies to a new level of safety and efficacy and more widespread adoption. Tim and his colleagues have been working on gene circuits for a long time, dating back to his time on the faculty at MIT. He left that esteemed academic institution to go to work full-time on turning this research work into cell therapies at Senti that will someday, hopefully, help patients with cancer and other diseases. Senti's work is still very early stage. It's all preclinical, but it plans to seek clearance from the FDA to begin its first clinical trial for patients with acute myeloid leukemia in 2023. Tim, like a lot of biotech entrepreneurs, is the son of immigrants. His story starts there and takes a few interesting turns before getting to this current chapter where he's running a company trying to push the frontiers of cell therapy. I think you'll enjoy hearing about the person and the science. Now, before we get started, a word from the sponsor of The Long Run. Calgary is home to more than 120 life sciences companies, from emerging startups to established firms. With this critical mass of research, technical talent, and expertise, the city is an active hub for life sciences innovation. Technologies homegrown in Calgary are changing the face of healthcare. Scientra is revolutionizing breast cancer detection using artificial intelligence-derived algorithms. Nanotest is harnessing the power of nanotechnology to tackle chronic wounds and skin conditions. And this is only the beginning. Calgary's life sciences sector is projected to spend $428 million on digital transformation by 2024. If you're a bright mind or bright company solving global health challenges, Calgary is the place for you. Take a closer look at why at calgarylifesciences.com. Now, please join me and Tim Lu on The Long Run. Tim Lu, welcome to The Long Run.
Thanks a lot for having me. Really appreciate the opportunity to be here.
So, Tim, I'm really excited to dive into some pretty hardcore science and technology here with your new company, Senti Bio. But just to get started, to familiarize the listeners, I'd like to ask a little bit about you. So, where are you from?
I was born in the Bay Area. My parents were from Taiwan originally, but migrated to the U.S. Pretty early on. And I spent time in the Bay Area and in New York and ended up going to school in Boston.
The Bay Area. Okay, so what did your parents do for a living?
My dad is in the semiconductor field. He actually went and did his PhD at Stanford. So I was born not too far away from there.
Okay. And your mom?
My mom's been basically working in accounting and business most of the time.
Okay. And you have any siblings?
Yeah, I have one younger brother. He's based out in Singapore and works in biotech now as well.
So how long were they settled in the U.S. before you were born?
Oh, that's a good question. I actually probably don't remember the exact timing. Probably a couple of years, I think. My dad came to the States for his PhD, and I was born during that time.
Oh, okay. So pretty recent immigrants. I mean, so they had to do the usual thing with learning the language, the culture, all that stuff?
Yeah. I think at the time there was not that many folks coming over from Taiwan. So from what I hear, it was certainly a transition period for them.
Okay. So this is sounds like an educated household. What kinds of values did your mom and dad seek to impress on you and your brother?
That's a good question. I think, especially my dad, kind of coming from an engineering background, had always encouraged us to really try to pursue something that we felt could have benefit for the world. And initially that was computer science and electrical engineering for me, and sort of just like following those footsteps. But pretty quickly, I realized that my own passion was actually in the biology space. So I actually made a transition over there, and they were super encouraging of that as well.
Yeah, yeah, I want to get to that. But what kind of student were you? Just like in elementary, middle school? Were you always like ahead of the class?
That's a good. Well, don't remember all the way that far back, but yeah, no, I guess I was, um, always doing decently in school. But I sort of like goofing around quite a bit as well, and playing sports. So it was sort of a mix of things. Things probably got a lot more serious for me when we got into high school, and then sort of preparing for college.
Uh-huh. What sports did you like to play?
A lot of tennis, play some volleyball, play some badminton as well.
Uh-huh. Uh-huh. Cool. So, what high school did you attend?
I was actually in the American school in Taipei. So actually, you know, about halfway through my time in the States, I was like in elementary school. My parents actually decided to move back to Taiwan. And so I ended up going back there with them, and had to learn a lot of Chinese from scratch. And then ended up going to the American school there for high school.
Oh, so what years were you in Taiwan?
In Taiwan, I think it was there 1990 to 1999. So about 9 years.
Oh, wow. Okay. So you're born and raised in the US, and you're kind of in that whole Stanford milieu. But now you're kind of straddling these two cultures,
Yeah.
getting a deep immersion in Taiwan. So did you like learn language and um, like, I mean, you must have had lots of classmates who were not all like Americans with your kind of background.
That's right. It was funny. I when I was in the States, my parents tried their best to keep me up to date with the Chinese language. I went to, weekend language school and everything, but it's really different when you're not immersed there. So when I went back, I think I started off at the fourth grade level for everything else, but my Chinese was at probably like at a pre-K or kindergarten level. So I actually remember pretty distinctly being set back and put into the class with the younger kids, so that I'd have to work extra hard and over the summer to sort of get myself out of that sort of situation. So it was pretty interesting. But I ended up really enjoying my time there. It was a completely new culture that I wasn't exposed to. So,
How was the school different than what you had been used to?
well, I mean, in the States, I went to a, pretty run of the mill public school. In Taiwan, I think, there's just a ton more emphasis on, on rote memorization and sort of hardcore drilling in terms of knowledge, which was pretty different than sort of the Americanized way of thinking. So it was there's certainly an adaptation, especially on the language side. I mean, for anyone who's learned Chinese, especially later on in their life, it's a lot of memorization that you need to do and a lot of practice. So that was certainly very, very new for myself.
Uh-huh. Uh-huh. So you're getting a little bit of left brain and right brain
Yeah,
exercise there. Okay. So you came back
yeah,
and you went to MIT. And I
that's right.
think you said that you started out in like engineering and computer science, but you decided at some point that wasn't really for you. How did you come to that?
Yeah, it was interesting when I first got to MIT. I mean, obviously a big tech school where, engineering is really, really a big focus there. And a lot of my friends and classmates at the time were pursuing, computer science and double A. This was around 99 or so. So this is, there's certainly a tech boom going on that sort of influenced everyone to go in that sort of direction. And it was fine. I didn't dislike what I was doing, but really what was quite exciting to me at the time was the Human Genome Project. Really started to come out and make a lot of news. And at the same time, I started reading some of the first papers around synthetic biology. They were started being published in the late 90s, early 2000s, really showing that you could build, switches, clocks, all sorts of really cool, demonstrations in living cells. And it was really fascinating because, when you start off in electrical engineering, you learn about sort of the basics of how computers work, right? You learn about how switches work, how clocks are made, how we use electricity to sort of do these things in our computers. And now, I started reading these papers about how the same thing was happening in biology, right? And it sort of just felt that, the sort of things that people were starting to do in biological systems was what I was reading about happened in the electric field, decades previously. So that was
Right.
really exciting. And, Boston happened to be, an epicenter of that sort of research at the time. So I got more and more into it at the time.
So now, but with engineering of the type you describe, I mean, it's got this mechanistic and deterministic way of thinking, and of course, biology doesn't quite work that way.
Yeah.
Can you say a little bit about like synthetic biology, like what it is for those who aren't that familiar, and why that intrigued you so much, coming from the things you had been learning at the time?
Yeah, I think that's a really good point, and probably points to a lot of the learnings that the field overall has made over time. I think early on, a lot of the concepts in the space were, people were trying to borrow analogies from what we knew, right? So, can we think about living cells like computers or like mechanical systems, and could we actually design switches and controllers and feedback loops and things like that in living systems? And conceptually, that makes a ton of sense. That biology would have feedback loops. We know like endocrine loops are sort of designed that way. And so conceptually, that's really interesting. I think the thing that is increasingly appreciated is obviously biology is not designed, by an intelligent designer. It's shaped through evolution. And so sort of the ground rules for how biology operates, like you said, are not built up in that sort of modular bottoms up, sort of first principles approach. And so to me, it's like, at the time, the field was really trying to start exploring whether those design rules were there, whether we could apply, true engineering principles. And I think it's becoming even more fascinating over time that in some cases we can apply those engineering principles, but in other cases we've had to start accounting for the complexity and interconnectedness of biology. And so it's really emerging as a new way of thinking about engineering when you're engineering a system that's really not human designed from the beginning.
So you made a switch. I guess, was this during your graduate school career, getting the PhD, where you got into biomedical engineering, or at least what was how that was understood at the time?
This is an interesting time to be at MIT because I think this is right about the time when the bioengineering department started getting formalization at MIT at the time. So I actually, as an undergrad, decided to add on biology and bioengineering as minors, and then ultimately, during my PhD, I made the switch over, and I did a lot of work in the engineering department, but really was focused on bioengineering as my main thesis project.
And now, what was your thesis project focused on?
Yeah, my PhD was around engineering bacteriophages, which are these bacterial viruses that we've known about for over a century now, but really trying to genetically engineer them so that they could be better treatments for infectious diseases and biofilms. So specifically, and sort of looking back, it seems kind of trivial what we did, but at the time it was a ton of hard work. We were able to take, a bacteriophage that infects, pathogenic bacteria and re-engineer them so that they could express things like enzymes to degrade biofilms or proteins that could suppress, the antibiotic resistance mechanisms inside of cells. And the idea here was hopefully this could be an interesting and useful sort of new type of antimicrobial agent that we would try to use more broadly.
Yeah, so these are basic techniques that could be applied in lots of different ways. Mm-hmm. Industrial applications, medical applications. Where did you think it had the most potential early on?
Well, I think early on, just maybe it was just the environment that we were in and sort of the ecosystem of Boston. Everything tends to be quite focused on the biomedical applications. So certainly we spent a lot of time thinking about, biofilm infections on medical devices. Can we use this to try to clear out infected catheters or treat antibiotic resistant bacteria in humans? I think the interesting thing about this technology is over time, we actually realized that this ability to engineer bacteriophage to infect bacteria, this is sort of a rudimentary gene therapy for bacteria. And so later on, the technology was commercialized and found some really exciting applications in the diagnostic space, using bacteria to detect pathogenic bacteria in food and other sort of directly sort of medical settings.
Okay. Okay. So yeah, you're talking about applications. You're part of the whole MIT, environment where getting your inventions out into the world and applying them is generally seen as a pretty good thing. Now, so when did you go on to the faculty? Did this like happen in a straight line for you? Like straight out of graduate school, you're a boy wonder or something?
It was I don't know if I'd characterize myself as like that, but so I ended up doing my MD PhD. I did it as part of the Harvard MIT HST program. So that was a really interesting time. It gave me exposure to the clinical side, and also to basic research. When I graduated, I was sort of open-minded about what to do next. Fortunately, at the time, I got a call from the MIT EECS department, Electronic and Computer Science. And they invited me to come in for an interview. I think the area of synthetic biology was intriguing to them, and so I actually at the time I don't recall actually applying to many faculty positions. This was sort of something that sort of came and ended up securing the job there. So I started at MIT in 2010, and it was a really exciting sort of launch for my own academic career.
But actually, in the computer science department.
Yeah, so I was appointed in electrical engineering, computer science, which is one department at MIT, and then I later on got a joint appointment in bioengineering.
Okay, so you get to straddle these disciplinary boundaries, if you will.
That's right. Yeah, that's right.
Okay, so now I don't want to go over like all your papers. I mean, the things you got to do in academia. You learned, you made progress there. And eventually you started working with industry. How did that happen?
Yeah, actually, my first foray into this was with my PhD project. So, with the bacteriophage technology, when we first started publishing those papers, it was late 2000s. We were trying to figure out what to do with the technology. We initially thought it made a lot of sense to try to use this for biomedical applications. But then, and even now, I think sort of the investor support for novel antibiotics or antimicrobials was still quite difficult. So together with a close colleague of mine, a fellow named Michael Karras, who was also in the same PhD lab, started looking at other applications for the technology. And that's where we ended up landing on, bacteriophages engineered to express a reporter that could be used as a bacterial diagnostic in industrial or food settings. So that led to the founding of a company, a spin out called Sample 6, that ended up moving forward and commercializing and developing, bacteriophage-based diagnostics for the food industry. So that was really my first experience with the translational side. And then after I started my own academic lab, was fortunate enough to interact with other investors, other entrepreneurs, and participated in the founding of multiple other companies where, as a scientific founder, was able to contribute some of the cool technologies we were doing in the lab and work together with, the investors as well as the management teams they pulled together to take those programs forward.
What was it about industry that attracted you? Because not everybody who, goes to join an academic institution, sees a need to work with industry.
Well, the reality of it is, if you want to move something forward, especially in the clinical space, the amount of resources it takes is quite high, and frankly, the barriers to get sufficient academic support, from government agencies, for example, to really move things along quickly is the barriers are pretty high in that sense. So, I've always been one primarily motivated by wanting to develop these technologies and hope they can make big impact on the world. But to be able to demonstrate that impact, we need resources. We need to be able to, manufacture. We got to go test these in humans, et cetera. And so industry has always been for me, a great outlet for trying to take some of those technologies and hopefully demonstrate benefit for patients.
Mm-hmm. Mm-hmm. One of the companies that I remember talking with you about probably 5 years ago or so was Synlogic.
Mm-hmm.
And this was engineering of live bacteria.
Yep.
Putting some circuits, as you say, into the live bacteria to give them different properties as therapies. Can you talk a little bit about like what the idea was there?
Yeah, the idea early on was that, we had been working to basically build what we call genetic circuits in bacteria because it was a lot easier than doing that in human cells. And so by 2014 or so, my lab, Jim Collins' lab, and others have published a bunch of papers on building bacteria that could sense, that could compute, that can make decisions. At the same time, you had a lot of growing interest in how microbes interacted with the human body, and it became increasingly clear that we had, resident microbes that could really significantly impact our physiology. So ultimately, the idea was, could you engineer some of those bacteria with novel functions so that they could act, at that host microbe interface to try to transform the treatment of diseases. And so that was the initial concept at the founding of Synlogic. It was a pretty new idea. Certainly people have been taking probiotic bacteria for many, many years, but the idea of being able to genetically modify them and turn them into a drug was pretty new. So companies come a long way in terms of developing clinical programs, manufacturing, and a lot of the translational expertise to really try to enable that new class of medicines.
Can you back up just a bit and say for our listeners what you mean by a gene circuit that goes into one of these bacterium?
Yeah. So, a gene circuit basically is a collection of genes that we put into a cell. And basically, we design those genes to interact with each other in a predefined way to give rise to like a specific engineered function. So, like a very simple example of this is a switch, where you might have two separate proteins, two separate genes that are expressing two separate proteins. Each of those proteins inhibits the expression of the other gene. You can imagine gene 1 producing protein 1 that suppresses gene 2, which makes protein 2, which suppresses gene 1. So it's sort of like this system where both genes are basically blocking each other. And so that sets up a system where either gene 1 is on and gene 2 is off, or gene 2 is on and gene 1 is off. That's a very simple example of a switch. For those, aficionados who know about phage lambda, that's, like one of the examples of how natural switches work, but we can make artificial versions of these. So basically a gene circuit is, a program that we can insert into cells composed of multiple genes. And really a lot of the art that has gone into synthetic biology is how do we design those genetic circuits to give us the functions we want, to do logic, to have memory, to sense and respond to a disease environment. It really comes down to how you program those genes and those interactions.
I remember the early days of Synlogic describing it as like, could you design a bacteria that would sense the changing conditions that it encounters in the body? So like you swallow it in a pill and it encounters this highly acidic environment in the gut, and then it needs to, migrate across the gut lining. There's going to, and could you perhaps, adjust the dose of the drug that you want to deliver when it goes from point A to point B?
Yeah, for sure. And actually, funny enough, we ended up publishing a paper, several years ago in Science where we showed that you could actually make bacteria that could detect, for example, bleeding, detect blood essentially. And you can actually build other types of sensors like that and actually have them work in an animal. And so there's sort of like three aspects of the way I think about these gene circuits. There's the front end sensing, like what can you detect, right? It could be, inflammation in the gut, or it could be an antigen on the tumor cell surface. Or then two, there's the internal computation, right? Is there some sort of logic? Only kill the tumor if it expresses A, but not B, right? So you can build that sort of functionality. And then there's the output. Like we can engineer cells to spit out, 1, 2, 3 different functions. They could be proteins that they're producing. They could, be granzymes to kill the cancer cell, et cetera, et cetera. So it's really that sort of sensing, compute, and then have an output that really is the paradigm by which we design these sort of genetic circuit elements. And that concept can be applied to a lot of different cell and gene therapy areas.
So you're already moving into this next arena. Like you started with the bacteria cells, and now we're talking about cancer. So around the same time, I guess, 2016, 17,
Hmm.
was when the first generation of CAR T cells
Yep.
were being developed and brought through the FDA. Very exciting moment for cancer treatment, that you could, design T cells to hit certain markers on cancer cells and kill them very effectively. So Novartis and Kite, now Gilead, were first there. So you looked at that with your background and said what?
Said a couple things. One was, holy, cow, that this is possible. That's amazing. And the durability and activity was really quite impressive. I think two, it was, wow, the time is coming, because back when we were doing research in the early 2000s, that was always the idea. And we were not the only ones, obviously. There's many pioneers working in the cell and gene therapy space for many, many decades, who tried to convince people that it wasn't a crazy idea, and to actually show that it could work safely and efficaciously. And so it was an amazing realization that we're now in an era where we could think about using cells as well as AAVs or gene therapies to try to treat diseases in fundamentally new ways. And this idea that you could actually use biology to treat biology, opened up the concept that you could actually start making more programmable medicines. I think the additional thinking was, wow, these first generation programs are impressive, but there's so much more we could do with these modalities. The first CAR Ts were really going after CD19, killing every cell that expresses CD19. The first gene therapies are just essentially overexpressing a replacement protein. That's awesome for certain diseases, but for most diseases, that's not sufficient. So in a lot of cancers, it's hard to find a clean target. Most targets are dirty in a sense. And so if I build a CAR T cell that only kills every cell that expresses a dirty target, I'm going to have a lot of toxicity on my hands.
Dirty in that these targets are expressed on the cancer cell, but also something quite similar on healthy cells.
Exactly. Yes, that's right. And so the realization was that, look, these modalities are here, like they've landed, it's exciting. There's so much more that we could do, but to reach for that next stage of a broader indications or greater efficacy or greater safety, we're going to need a lot more improvements, especially at the genetic engineering level. And that's really what synthetic biology has been trying to develop for many years. So it was sort of, is it the opportune time to try to put these platforms and approaches together?
And everything was there, like with the CAR T cells. I mean, they were doing this in the lab. So it's this controlled environment. You do the engineering of the T cells. They're reinfused into the patient. There's a window of opportunity to do some other things to those cells.
Exactly.
Okay. So by this point, you had been involved in starting a couple companies, as we discussed. How did the idea for Senti come about?
Yeah, so I had a really good MIT classmate, my co-founder, Philip Lee. We were undergraduates together. And we had always, he had ended up going to do his PhD at UCSF in Berkeley in the bioengineering space, and went more on the device side. So he started making microfluidic devices that could, manipulate mammalian cells. Eventually started a company called CellAsic that was acquired and by a larger pharma company. And then he worked there for many years, leading their cell cultures technology business. He and I had always kept in touch and really were observing sort of the rise of enabling sort of manufacturing technologies for cell therapies along with synthetic biology. And so we started, reconnecting after some of these clinical successes came about, and started brainstorming about, was the time right? Could we pull the technologies out of, the academic labs and make a platform company that was able to design gene circuits specifically for human cell and gene therapies? Some of the concerns that we had at the time was, was it too early? Was the manufacturing technology available, et cetera, et cetera? But we felt that, ultimately it was the right time for us to give it a shot. That, we felt that this is where the field was certainly going to go. And there was a major opportunity to try to bring these technologies out from the lab and try to make them see the light of day.
Now, but you still had a day job as a professor at MIT. What was your thinking here? Like that you could you remain on the scientific advisory board and do that kind of arm's length thing that is commonly done, or go all in and be the full-time CEO?
It was interesting. This is right about the time that I was tenured at MIT, fortunately, and usually that comes along with having a sabbatical. So I spent a bit of that sabbatical time, formulating and started Senti with Philip. That gave me, at least personally, a little bit of time to sort of evaluate and see if this sort of new world was what I wanted to do.
So when they're trying to put their hooks in you with tenure, you're about ready to jump ship.
Well, I would say that, at MIT, if you work for 6 years, you're obligated. You're guaranteed your sabbatical. So it was part of the deal early on, and there was no exceptions made for me personally at the time.
Okay, so you're sketching this out with Philip during your sabbatical. Got it.
That's right. Yeah. So we started working together quite then. It's been fun ever since.
But you decided to why did you decide to go all in and even like move to California?
Well, Philip was based out in the Bay Area, number one. So there's sort of a personal need to sort of like be together and start this thing. But I think the reason why I personally, decided to jump full on into Senti was, and this goes back to why I started in the field in the first place. Like I started in this field because ultimately I hope this is, just the beginning of this revolution, right? Semiconductors, the first transistor, nobody knew how far that was going to go. Look where we are today. I have the same feeling about biology. We're just starting to unravel the complexities of biology. We're still in the very, very early innings of being able to engineer biology. And I just felt that, for me personally, I wanted to see that be able to make the next step. Like, can we actually show that these technologies not only are cool science nature papers, but can they actually really help humans? And I think the realization was that, engineering these sort of genetic circuits, especially in human cells, was really challenging. It required a lot of sort of accumulated mistakes that one has made to learn about how to do this well and as efficiently as possible. So for me, it just felt like, I had to go all in on this if I was going to make an impact and try to see this one through.
Calgary is home to more than 120 life sciences companies, from emerging startups to established firms. With this critical mass of research, technical talent, and expertise, the city is an active hub for life sciences innovation. Technologies homegrown in Calgary are changing the face of healthcare. Scientra is revolutionizing breast cancer detection using artificial intelligence-derived algorithms. Nanotest is harnessing the power of nanotechnology to tackle chronic wounds and skin conditions. And this is only the beginning. Calgary's life sciences sector is projected to spend $428 million on digital transformation by 2024. If you're a bright mind or bright company solving global health challenges, Calgary is the place for you. Take a closer look at why at calgarylifesciences.com. I covered these companies, around this time with the first generation T cells. And as you said, they were targeting CD19. Mm-hmm. It's that antigen expressed on B cells. And you could make these engineered T cells that reinfuse back into the patient, and they just kill everything that carries CD19. Yeah. And it's okay. Like, it's not going to kill the patient. It's going to kill the kid. Well, I mean, there were side effects that were other things that people found out about later. But depleting CD19 was not bad. Like you say, there were other antigens that people had on the drawing board that might be useful, but were not approachable. And you thought maybe this kind of more nuanced circuit, if it could be engineered into the T cell, could give you the balance of the efficacy and the safety that you need.
Mm-hmm. Yeah, and I think, this is still something we got to prove widely, obviously, as we move forward into the clinic. But I think the realization is that biology actually kind of acts this way already, right? Like the NK cells that we have in our body, they have activating receptors and inhibitory receptors. And the way they go about the body and try to figure out, is this cell infected by a virus, or does this cell look weird from maybe it's an early signs of a tumor, I should kill it, is by sensing using these activating inhibitory receptors, how strong of a signal am I getting that this thing is abnormal? Similarly, we know that biology is made up of feedback loops, right? Our whole endocrine system is all about, producing hormones, sensing the amount we're making, and then sort of correcting for how much we should be making. Our whole circadian rhythm is based on clocks. So this idea that quantitative functions inside of biology has become increasingly clear over time. The idea that the genes are static, you knock one out and you just see what it does, or you overexpress and see what it does, was very powerful in the early days of genetics. But biology is much more complex than that. So I think the hypothesis has always been that if biology sort of acts that way already, why don't we have drugs that are like that? Most of the drugs that we take today are sledgehammers. They target a single thing, they hit it really hard, and they go away. Can we create drugs that are more controllable, more nuanced, more specific? I think we still have to demonstrate that this is going to be a major new modality, a new class of medicines. But I think the scientific rationale for why we should have medicines like that, to me, is very strong.
Now, there are different tools that one can use to achieve this similar kind of objective. People have heard of CRISPR, right? Why might you prefer to use CRISPR in some circumstances, or why might you prefer a synthetic biology, circuit, in another?
Yeah, it's a great question. I don't actually think of CRISPR and synthetic biology as conflicting with each other. The way I sort of think about it is, let's say I'm trying to write a story. There's a lot of tools I can use to write that story. It could be someone gives me an existing story and I take a pencil and an eraser and I erase some of the language there and I change the language and I change the meaning of the story. Or I sort of sit down in front of a computer and just like type it out from scratch and create something completely new. And depending on what your goals are, if you're an editor or if you're a novelist trying to create something from scratch, you may take a different approach. They really have different use cases. And I think that's really similar to how genome editing interacts with synthetic biology. For diseases where we know there's a single gene or a small number of genes that are responsible for the phenotype, there's some error in those genes in the human because of some genetic background. Going in with a CRISPR element that can fix it, make a small change, and restore it to the quote unquote healthy state makes a lot of sense. It's a very direct way of treating diseases. But there are other diseases where it's hard to find that single driver, or there's biological limitations on you can't gene edit every single cancer cell very easily, where you have to take an alternative approach where we're need to use cells or need to use viruses as the therapeutic modality. And there we need to actually fundamentally reprogram what the cells are doing. If an NK cell or a T cell was naturally able to cure cancer all the time, we would never have cancer. So clearly there are some missing features in the way our bodies and our cells operate. And if we can take advantage of those cells, reprogram them, insert new functions that are sort of an upgrade on what they do naturally, that's another alternative way of trying to treat disease. So I think each one of these technologies is going to have a very different place in the treatment ecosystem.
So you've mentioned a couple different cell types. So there's a lot of different directions that you can go with these circuits. There's NK cells, there's T cells, there's hematopoietic stem cells, the ones that form blood. There's other things you can do too. How do you decide where to concentrate your energies?
Yeah, that's a great question, and one that we ask all the time at Senti. I think stepping back, the cool thing about what we've built here is really a platform that can design gene circuits across many different applications. And it actually turns out that like the genetic circuits you might build for one cell type, oftentimes can actually work in another cell type. They might not work the exact same way or as efficiently, but the basic function usually is there. So there's actually a lot of cross-learning that you can gain from keeping this all under the same roof. So early on at Senti, we created a core synthetic biology gene circuits design team, where this team does all of the designs, whether it's with our internal programs or with external collaborators. This has allowed us to really gain and learn from each round of experimentation. When we go around the design cycle, we design, build, test, and then we learn from that. When we fail, we make improvements, and that improvement gets stored in our central sort of knowledge database.
This is where you're designing the constructs, the nucleic acids, which are the code. And this sounds kind of like software style. Exactly. I mean, you got to be careful with the analogy here, but there's sometimes you write lines of code and they're bugs in it, and you revise and hopefully you improve it. You run it through an experimental model and see if you fixed it.
You got it. Yeah, it's exactly how we approach engineering. I mean, we can talk about some of the nuances to this later, why it's not purely as bottoms up as we would like, but certainly that's the approach we take, right? So the more things we can create in parallel, the faster we can test them in our experimental models to get to a yes, no, does this work or not, the better we get at doing this over time. And that's really the ultimate sort of hypothesis behind Senti, is that we want to be this accumulated knowledge database of how to design these. Now, from a downstream clinical development perspective, there is the reality of, clinical trials are lengthy and expensive, and there's only so much a small company can do. So we made the internal decision to focus our pipeline in oncology. We felt with the high unmet need there, as well as some clear reasons for gene circuits to exist, we could potentially design trials that go in and really show benefit for patients. And that's really what we're driving towards. However, outside of oncology, we've chosen to partner with other great teams and great companies. So we have a partnership with Spark in the gene therapy AAV space, where Senti is designing the promoters, sort of the software that controls when and where genes are expressed to try to achieve much more specific targeting. We also have a collaboration with Blue Rock around iPSC engineering, building our gene circuits in that context. So we do want to continue to drive forward our own pipeline, but we also want to be, really a partner of choice and a collaborator with anyone who's interested in making that next generation of cellular gene therapies.
So on one side of what you do, you can sort of widely out-license your technology for making these constructs, the code.
Mm-hmm. That's right. Yeah.
But
so
then the rest of it, you're going to keep in-house to develop your own therapies.
For I think specifically around NK cells in oncology, which is where we're focused on now, at least to date, we've chosen to keep all of that internal. I think outside of oncology, there's certainly a lot of rationale for collaborations with other groups.
Now, you've mentioned NK cells a couple times. Sounds like this is a priority. Why have you focused that energy on NK cells? And I think these are the allogeneic or off-the-shelf, non-personalized kind of NK cells.
Yeah. The interesting thing is, several years ago, we actually showed that our, logic gates, our switches, et cetera, could work both in T cells and in NK. So from a technology perspective, both of those are equivalent in our minds. I think the exciting thing about NK cells, especially over time, as more clinical studies have been done, is number one, they seem to have this, improved safety profile. They can enable, allogeneic off-the-shelf therapies, given that they don't generate sort of the issue of GVHD. And so from a product perspective and thinking about making this into a real drug, we're really excited about this particular cell type. There are some challenges, certainly with NK cells, but we think we can address actually a lot of those challenges through the gene circuits that we're actually building now.
So I sort of glided past the side effects. You brought it up again, but yeah, there's been issues with CAR T cells, the first generation ones, with triggering cytokine release. Mm-hmm. So this is where the patient mounts a really massive inflammatory response that can be dangerous because basically the T cell is so good at killing the tumor so fast.
Yeah.
That's a reaction that can be dangerous, but then there's also a neurotoxicity, and there are lots of companies that have been working hard on that. So maybe did you see, like, okay, other people are kind of on those issues and trying to engineer the T cells to take care of those issues, and maybe we've got some other more green fields open to us?
I think it was more driven by the idea that NKs, at least on their own, even without a lot of engineering, seem to be less susceptible to those similar T cell issues. So from a cell type choice, could we start with a cell type that had this more benign profile and then do our engineering on top of that, is sort of one of the thinking. The second thing, which is a more practical one around manufacturing, is sort of the autologous manufacturing work stream. We have to take the patient's own cells, genetically modify them, and get them back to the patient really quickly. Imposes certain constraints on how much you can do with those cells from an engineering perspective. And so the ability to take NK cells or other allogeneic cells from a healthy donor, genetically modify them, expand them, cryopreserve them, has a lot of sort of nice features associated with trying to make an off-the-shelf product, especially when we're starting to do more and more sophisticated genetic engineering.
Okay. Can you talk a little bit? So this is one thing I really wanted to ask you, because last time we talked, you were still a private company and playing the cards pretty close to the vest. And now that you're a public company, you've had to put a few more cards on the table, like talking about what's in your pipeline, your top programs. You presented some data last fall, I believe November, or in advance of the American Society for Hematology meeting, about now this is all still preclinical. These have not gone into patients yet. But could you describe that lead program that targets FLT3 or CD33?
Yeah. So this is a program we call Senti 202. It's really trying to address one of the key challenges in AML. In AML, we think there's two big problems that we were sort of faced with. One is there's no single antigen, as far as we're aware, that hits all of the AML cells. Many of the antigens that people have gone after may hit the AML blasts, but may miss the leukemic stem cells or vice versa.
So this is acute myeloid leukemia, still one of the hard to treat blood cancers.
That's right. Exactly. Thank you for helping to clarify. So there, you have this issue of essentially antigen heterogeneity in the AML context.
There's no single antigen like CD19 that applies here.
That's right, exactly. So you can't just go in and blast everything that has that antigen. The other issue is that a lot of the antigens people are looking for in acute myeloid leukemia are not just expressed on AML cells, but they're also expressed on the healthy cells. So if you have an agent that goes in and is killing everything that expresses that target, you're going to have a lot of toxicity against the healthy tissues in these patients. And as a result, you can't hit the tumor as hard, you can't treat it as aggressively as you'd like, and you're worried about toxicities. So to us, this is a really good example of one of the central problems that I think oncologists have been dealing with ever since we knew about cancer, which is how do I make new therapies that basically tell the difference between a cancer cell and a healthy cell? Like, how do I get that pinpoint accuracy? Because if I can, then I can throw all the toxic, all the killing at the tumor while still sparing the healthy tissues. And so that's really hard to do with a conventional sort of single target. If that target's dirty, it's what limits, ADCs, antibody drug conjugates or bispecifics, again, if you only have a single target. So what we realized pretty soon is that cells don't have to be necessarily limited in this way. We could design cells that actually detect multiple targets at the same time and then make a decision about what to kill and what not to kill, depending on which targets are present. So in Senti 202, we actually have two major components. The first is an activating CAR. So there's a chimeric antigen receptor that recognizes CD33. It also recognizes FLT3. So these are two pretty well-known tumor antigens that have been, used in AML before. And so engaging either CD33 or FLT3 will trigger killing of the cancer cells. The benefit of this is, because we can kill anything expressing CD33 or FLT3, it actually creates a broader spectrum of killing overall, and we believe will allow us to expand the different types of AML cells that actually would be treatable with this sort of approach.
And is there a lot of diversity among these AML cells? Do some of them carry CD33 and others more FLT3?
Exactly.
But not usually both?
Sometimes they have both, but oftentimes you'll find that some are more CD33, some are more FLT3, and it's actually quite diverse between patients and also within patients. So there are some, if you take a patient's AML sample, and you look at all the AML cancer cells, some of them may be more CD33, some of them maybe more FLT3. And so if you only went after one of these targets on its own, you would miss that population, unfortunately. And that would potentially translate into tumor relapse.
Okay. And your NK cells, they also have this code in the construct that says, don't target EMCN.
That's right. Yeah, exactly. And so that's the part that we're especially excited about, because I think this would be a key differentiator in the clinical setting for the treatment of cancers. So what this is what we call a NOT gate. It's a NOT gate because it basically tells the NK cell, do not kill this particular tissue or cell type. So it essentially acts as a brake on the treatment. The way it works is basically we identify what we call a protective antigen. So a protective antigen is a protein that's highly expressed in the healthy cells, but not expressed in the cancer cells. And then basically This NOT gate is designed to recognize that protective antigen and basically, tell the NK cell to stay away from these healthy tissues. And so if we can do that, we think we can protect the key healthy population that we're worried about in AML, which is the hematopoietic stem cell. But we also think this sort of approach applies to many, many other tumors that are out there, especially solid tumors, where, again, it's very hard to find a clean target. And we want to try to minimize toxicity against key organs, in the heart or in the lung or in the gut, et cetera, and prevent that toxicity that we see, which hopefully will translate into the ability to treat these diseases much more aggressively and effectively.
Now, this marker EMCN, it's on the blood-forming stem cells, as you say. So that ought to protect the, it ought to help the patient avoid the depletion of the white blood cells that makes you vulnerable to infection, the depletion of the red blood cells that makes you anemic. These things that are well-known side effects of more blunt instrument cancer treatments. You're trying to spare those cells. That's
right. Yeah, exactly. We're trying to spare those healthy cells. And it's that concern over, what we call on-target, off-tumor toxicity against, for example, those stem cells that oftentimes limits, how aggressively you can dose or how aggressively you can treat patients suffering from cancer. And so our hope is that we can decouple that issue from, sort of the treatment of the tumor cells themselves. Basically, can we protect those key healthy cells and still allow you to hit the tumor as hard as you can? Now, do these constructs come with limitations? Because, I can imagine you got to have kind of like short and sweet, elegant code as best you can so that there's not a lot of, extra crud in there. Yeah, that's right. Just like you write code in a computer, you don't want to create spaghetti code that's too long unnecessarily, and you got to test it. So, when we're designing these gene circuits, we go through many, many, many iterations. You can change every single base pair you want, and there's, infinite combinations that you could change. So we need to test enough that we're confident that we picked, the best constructs to move forward. So that's certainly an engineering challenge. The other thing that's related is manufacturing, right? We need to be able to deliver these into cells, make a high quality product, et cetera. And in that context, generally speaking, the smaller the overall gene circuit or the code is, the easier it is to do the manufacturing. And so we spent a lot of time looking at how do we get rid of extraneous elements if they're not useful? How do we encode things in a way that's more compact to really try to, make the lives of our manufacturing colleagues easier? What's the size that you have to work with? How many kilobases? We're using viral vectors to transduce our NK cells. So, there's no hard upper limit, but we're certainly looking to try to, keep things within, 8,000 or 9,000 base pairs or less, ideally. Okay, so you use the viral vector to perform that reprogramming on the patients. Oh, well, in this case, these are allogeneic. So these are NK cells that you make, and you'd run through some test validations to make sure that they're doing what you intend in the lab dish before they were to go out and be put into patients. Yep. How far away is this from reaching your first real test in the clinic? We're targeting INDs next year at this point. INDs in 2023. That's right. Okay. Can I ask you a little bit about the business too? Sure. So I know that we talked about this this lead product candidate that you teed up there at ASH last November. Around that same time, you sought to do a SPAC deal, and to bring in extra money to invest. What was your thinking about what would that transaction enable you to do? Yeah. So, one of the reasons we obviously finance through any means is to be able to advance these programs forward. And, because, in the cell and gene therapy space, sort of the surrounding infrastructure for manufacturing these products, taking them forward into the clinic, analyzing them, et cetera, it's still a relatively new and developing field. We've felt that it's quite important for us to really build up the internal capabilities around process development, to be able to know that we're making high quality products and taking these forward. So the financing that we brought in, which we're very proud of and happy to bring in, over $140 million of gross proceeds, allows us to plan for that future and to build up the internal expertise and know-how to be able to advance Senti 202 as well as our other NK programs forward towards the clinic.
And this is important. You have some expenses to do this the way you want. Like with that rapid design, build, test, iterate whole thing, it could, I would imagine, slow you down if you had to rely on a contract manufacturing organization for every single one of these tweaks. So is this just a matter of like having more speed, more control, own it in-house? But the trade-off here is that you're going to have to spend more money to create that capability.
I think the way I would think about it maybe is a little bit differently. There certainly are great contract manufacturers out there, but I think for us to be, a great drug developer, whether we're doing it internally or working with those contract manufacturers when it makes sense, we need to know internally what exactly we're looking for. I think the model that doesn't really work in this field is simply relying on an external provider without really having a really good sense of what needs to be done technically. Because then, you can lose time and lose insights and lose control of the process. So I think an important thing for us is really building that internal know-how and oversight over how to manufacture our candidates. And in some cases where it makes sense to use a contract manufacturer for some of the components or some of the elements, certainly that's part of the consideration. But I think it's all grounded in having that internal expertise to know what good looks like in the process. So I would certainly not rule out the use of contract manufacturers. I think it's more about having that internal expertise to make sure it's being done the way you want it to.
But your product isn't just the code underlying the circuit. It's the cell itself, everything that goes into making the cell. So you do need to control that and have intimate knowledge of your own product.
That's right. Yeah, I totally agree.
Okay. So, but now, I mean, there was a little bit of business story here in the spring. Of course, the biotech markets were down. That's been told quite well. The SPAC deal took a little longer, I think, to close than would have been ideal. Looked like you kind of ran low on cash for a while. What did you have to do to adjust before you closed this thing?
Yeah, it's a great question. I think the thing I've learned about this, running a company, both private and public now, is we can do the best that we can internally. We can't really control the external forces. So I think certainly, with the funding that we had and the ones that we're using going forward, we're really trying to be very prudent and creative about how we spend money. So we have always looked at trying to do things the most efficient way possible and try to manage things through. I don't think we're cutting corners in terms of any of the sort of important work that we're doing, but there's certainly things you can try to do more efficiently in terms of the processes, the experiments that you're running, how broadly you run them, et cetera. And that's something we try to take into account on a day-to-day basis so that we make sure we're keeping the company in good financial shape and really driving towards the ultimate goals. I think the thing that I'm most proud about is that we have a great group of scientists and engineers here. We're just super passionate about creating this next generation of medicines. And so at times when we've had to, ask our colleagues to work harder or to be flexible or to think creatively, I think people have always risen up to the challenge. That's part of the culture we tried to engender here because we don't know necessarily what the future is going to hold at this point.
Well, and every company in biotech, if you're around long enough, is going to run into a rough patch. I mean, this just is how it works. Whether it's external or something that throws you a curve in your own development, it's going to happen. How do you seek to, keep the morale up, keep your eye on the prize, and encourage people to do this thing, which is really unprecedented. It's hard.
Yeah, that's a great question. I think number one, when we try to hire people, I think generally we're looking for people who are intrinsically motivated about our mission here. Because I think what you said, what we're trying to do is a moonshot in a sense, right? If we're successful here, it's going to blow open a whole new class of medicines and approaches. And so it's going to be hard. I think we don't try to hide that fact from people. And so in a sense, we tend to find people who are just passionate about that mission, number one. I think two, it's really keeping the eye focused on why we're here. And, I think people here believe that these products have the potential to really change the treatment of cancer. And a lot of people here have been touched by that. So, we've spent time, talking to the patient advocacy groups, bringing in, patients to really talk with us and really making sure people are really bought into the vision. And then thirdly, as management, we try to do the best that we can to make sure that people are well compensated and supported in their work, and try to provide as good of an environment that we can in that sort of setting. But I think it's really comes down to the intrinsic motivation. Like, why are we here? We're trying to create impact. We live in the Bay Area. There's other ways of making a quick buck that one could do, but it's more about we think it's really hard to do what we do. There's not that many companies that have the potential, and so if you want to be part of that story, have the Senti.
Have you read something lately, like a book, or I don't know, had a conversation with a peer CEO recently that you found inspiring and kind of helped you think about navigating this territory?
Well, I certainly have had many of those conversations, but probably one of the books that I read early on, which is really about Amgen, Gordon Binder's Science Lessons book, really talked about sort of the tumultuous times that they went through. And I think it was, I drew a lot of analogies from that, in that they were creating and being involved in really new classes of protein medicines at the time. And it didn't seem that different from the sort of environment that we live on today. It's sort of, there's ups and downs, like you say, in all businesses. And it's really about sort of seeing the long term and trying to encourage the team. I know a lot of my fellow peer biotech CEOs and executives are doing the same thing within their own companies. And I think one of the fortunate things of being in this field of, science and biotech is, most people are here because they want to make that impact longer term. So we have sort of that additional sort of thing to talk about with our team members.
Well, Amgen is now part of the Dow Jones Industrial Average, been around 40 years. It wasn't always that way. I know, I don't know, I'm sure that in that book, I have not actually read that book, but I know one of the stories is that when George Rathman, the original CEO, was there, he invested in a factory for making EPO before they got FDA approval. That took some guts, and it turned out to be a prescient decision. So I don't know if there's a lesson there for today's leaders thinking about the future of manufacturing. But
yeah, I think a lot of this comes down to what you think will come and making a bet on it and seeing if that bet turns out right. I think manufacturing certainly is important in the cell and gene therapy space. Don't get me wrong about that. And it's like you said, sort of having good control of your process is ultimately the most important. I do look forward, hopefully, to one day when there is a more robust manufacturing ecosystem around cell and gene therapies, because I think it's actually only going to help us accelerate the sort of things that we're doing at Senti ourselves. If we could focus on designing the software and have, another group that's really focused on the manufacturing, that's going to make sort of this design, build, test, and ultimately learning cycle, much more efficient. But that's not the ecosystem we have today.
Yes, but I think it's beginning to evolve that way. You familiar with Resilience? Their CEO was a previous guest on this show. I know Elevate Bio is another one providing services. There's a maturation, I think, of the cell therapy manufacturing ecosystem that'll make it a little more easier for innovators like Senti to plug and play.
For sure. Yeah, we're looking forward. We're looking forward to a lot of their hard work in that space.
All right. Tim Liu, thanks so much for joining me.
Thanks so much, Luke. Really appreciate it. Thanks for the invite.
Thanks for listening to The Long Run, a production of Timmerman Report. Pedro Rosado of Headstepper Media was the sound editor. Music is from D.A. Wallach. See you next episode.